In talking to my friends who are scientists, I think we all sort of feel that sense of wonder, that sense of discovery, this feeling that even the most trivial thing that I'm doing in the lab, I might be the first person ever to know this little piece of information, and it feels really interesting. It feels really fun. It's like I'm peeling the onion. I'm peeling back a little bit more of the truth. I'm trying to learn about, how is our world working.
I have a lot of ideas of different endeavors I want to pursue. I think it's my personality a little bit, always need to be on the move. I think as a scientist it's good to not always be in a comfort zone. As the first time I heard about CRISPR was around 2006, when I read some articles explaining that CRISPR could be used as a way to classify in a more, let's say, fancy manner, different strains of bacteria. So this was the first encountering with the CRISPR systems. Then I read about it, and that was it for me.
I think I first heard about it from Jill Banfield, my colleague here at Berkeley, sometime around 2006. And she was working on bacterial genomes and sequencing bacterial DNA, finding lots of examples of these repetitive sequences called Crisprs. And hadn't probably paid a lot of attention to them until some research that was published that showed that those sequences included little snippets of viral DNA. And this, was very intriguing because it suggested that bacteria were, keeping these little copies of viral sequences around for a purpose. Some research suggested that CRISPRs were using these little RNA copies of DNA sequences to detect and destroy viruses.
I was, at the time, a postdoctoral fellow in the lab of Jennifer Doudna, kind of fresh off the boat. This was at the time when Rodolphe Barango's science paper came out, establishing the CRISPR system as a kind of a primitive bacterial prokaryotic immune system. So my first exposure to this was at a journal club that we used to have in the Doudna group. I remember at the time we were, speculating how could this possibly work.
The knowledge was really limited, so at the time what was hypothesized is that CRISPR was a mechanism that would allow to recognize an infection with a virus and ultimately kill the viruses by targeting the genome of the viruses using protein and RNA components. So this was the hypothesis in 2006.
In Puerto Rico in the spring of 2011, I met Emmanuel.
We were giving talks at the same session, the same CRISPR session.
She said, I've been wanting to talk to you. I'm glad to meet you in person because I've been thinking that you might like to collaborate with my lab on a project. We were talking about a protein that at the time was known as CSN1 and now is known as Cas9.
This mysterious CSN1 protein, surely we were predicting the function, but no one could show up to now how it was really working.
We had this idea that, okay, the Cas9 protein was somehow going to be cutting DNA, but how exactly it was going to do it, we didn't know at the time.
And for Jennifer Doudna was extreme, let's say, curiosity on really the details of how those proteins work.
At the end of the conversation, she said to me, I think it's going to be really fun to work with your lab on this mysterious CSN1. And when she said that, this mysterious CSN1, I just felt this little chill, going down my back, thinking, this really is going to be a very interesting protein to study.
The collaboration ultimately went very, very fast. Involved a PhD student from my lab, Krzysztof Szylinski, and a postdoc from our lab, Martin Jinek.
Our goal was to really find out what the biochemical function or the mechanism of this Cas9 protein essentially was.
Right, somehow RNAs seem to be cutting.
What we do as biochemists is we set up systems in the laboratory and ask, in isolation, what does this particular molecule do? What is its function?
If you do the experiments with the components purified, you bring them together in a test tube and you show that it's working, then you know that you don't need anything more. It's self-sufficient. It's working like this. You don't need anything else from the cell.
We said, well, let's see if we can take that strategy using purified Cas9 protein, and we also made purified Crispr RNA molecules, these little RNA copies of DNA sequences, and then combined them together, and then started testing to see, can it use the letters in the RNA of that CRISPR molecule to find a unique DNA sequence and cut it? And initially, the answer was no. No dice. Nothing working.
This is a process of trial and error.
There could be some silly technical thing going on, like, our protein purification isn't any good, or somehow our sample that we're testing is just, has been destroyed in some way. Or maybe there's something missing from the reaction. There was a second type of RNA that Emmanuel's lab had been studying as part of this CRISPR system called tracer.
Tracer RNA. It was an RNA molecule which we realized was very abundant in the bacteria. Most likely there was an interesting function. And we realized ultimately that this RNA was encoded by a piece of DNA that was in the vicinity of the DNA coding for the CRISPR system. Maybe this tracer RNA molecule has something to see with CRISPR. Maybe CRISPR needs other components. So I remember that the students of my lab were telling me, no, this is not the dogma. CRISPR needs the CRISPR proteins and the CRISPR RNAs. You don't need anything else.
But we started thinking about it and wondering, maybe it would be important to include the tracer RNA. And this was actually an experiment that was first done by Krzysztof Chylinski, the student in Emmanuel's lab, who was our collaborator on the other side of the big pond. And he did that experiment and got a beautiful answer, which was that yes, when you include the tracer RNA, the CRISPR RNA, and Cas9 together in this reconstituted, purified system, now that Cas9 protein could find a unique DNA sequence and cut it.
This was one of the milestones in that project, realizing that this tracer RNA was in fact also required for the process of cutting the DNA. Once we knew that, then of course we wanted to see what parts of the tracer RNA are needed for the process. I did a set of experiments that suggested that certain parts of the two RNA molecules were essentially dispensable, but they were dispensable in a way that would allow us to fuse the two RNAs together and in doing so reduce the complexity of the system. Just go directly to DNA cutting without having to put these two RNAs together first somehow. This then led to these experiments with these single RNA guide fusions.
Martin had made several versions that differed just in the letters of the sequence that would allow Cas9 to find a different piece of DNA.
By basically designing the sequence of the guide RNA, you could redirect it to essentially any sequence in double-stranded DNA.
So we had this thing programmed with 5 different RNAs, separate samples that would find, in principle, 5 different places in the DNA and lead to a cut. And we were in the lab looking at the result of this experiment. We're leaning over the light box, and we could see that each piece of DNA was a different size, corresponding to the length predicted based on the ability of these single guide RNAs to direct Cas9 to cut the DNA at a particular place. We had been able to engineer this thing to be different from how it is in nature and much simpler in a sense.
It was a great moment. This was going to be a programmable system that you could repurpose for other things, including genome editing.
Here was something that was truly, fascinating. First of all, just fundamentally about nature, and then thinking about how you might be able to use that in other settings and other kinds of cells was also incredibly fun, because the more you thought about it, the more ideas would come, because it was just clearly going to be so useful for a lot of things. So there was a very intense few weeks finishing up the last few experiments and writing up a manuscript.
And when we were writing the paper, actually, the bright days were coming in Sweden. It's very disturbing physiologically speaking, because you never see darkness, you just see like a gray sky. I would work, I would have a tendency to work a little bit around the clock and to not sleep a lot.
The excitement was building up. So the final weeks in this process, there was this, playing ping pong basically with the manuscript.
I would realize it's 3 a.m. in the morning because I was corresponding with California, and realizing that no, it was really time maybe to get some hours of sleep. And ultimately, the paper that was published in 2012 in Science, showing the biologists how they could harness the system, so providing them the guidelines, the recipes to use the system for their own purposes, which they did successfully, because 6 months after the publication of our paper in Science, very fast we saw publications showing that the technology was working for genome editing and even engineering in plant cells, in human cells, in mice, in different model organisms which biologists use. So this was very, very fast actually.
It was clearly going to be a race, because if it worked, it was going to be so exciting. I wasn't thinking about, oh my gosh, I mean, this is a tool that, it fundamentally allows us to change our relationship with nature. It actually allows us to change human evolution if we want to, right? It's that profound. And it puts into all of our hands as human beings a tool that, we've never had in the past the ability to change the fundamental chemical nature of who we are in this way, right? And now we do. And what do we do with that?